Welcome back to the course on nanotechnology and its role in agriculture. So, the previous class we started with iron pyrite and we talked about the different role of iron pyrite for last 100 years or a little more than 100 years. And if you recollect, so we talked about the ubiquitousness of iron pyrite. So, it is kind of available most of the places, and it has been used for, or there are attempts are being made to use it as a photovoltaic material, and in India it has been largely used during 1970s, 75, 78 to reclaim sodic soil or rather alkaline soil, because once it gets into the soil in bulk amount, it make the soil pH slightly lower. So, from an alkaline soil you can bring it down to a neutral pH by adding iron pyrite, and it has been observed in such soil it has been led to more nitrogen absorption by the crops, especially by the rice crops. Okay. Whereas, when it has been used directly into normal soil, it has actually made soil acidic. So, one has to really handle for any kind of soil reclamation, it has to be handled with extreme care. Okay. Apart from it, it has been observed that these kind of material has something called a stoichiometric defect. What is a stoichiometric defect is, that is where we ended in the last class. So, this is where we are going to talk about a stoichiometric defect. So, whenever we talk about FeS2, the assumption or understanding is that there is one atom of Fe, and two atoms of sulfur. Now, in real life, that is not what happens. What essentially happens is, out of say 10 million molecules of FeS2, the surface there will be molecules which, or maybe slightly into the core, there will be molecule which does not balance it out, like all the time the ratio 1:2 of iron to sulfur is not maintained. There are molecules which may not have one sulfur, maybe instead of FeS2 in the cluster there will be some Fes. So, likewise it has been observed that the final ratio what it turned out to be is 2 minus x, and this x could be 0.5, 0.2, or it could be 0.1, whatever, that is immaterial. The point is not that every iron atom is bonded to 2 sulfur. There will be iron atom which may be devoid of it, and these kind of defects are called stoichiometric defect, and a stoichiometric defect plays a critical role in the surface property of these molecules, and this is what is very critical for you to understand, that iron pyrite comes with set of stoichiometric defect, and that is why lot of its application in electronics kind of got hampered because of it. But there is a positive side to it, and we will come later into that. What is the positive side of having a stoichiometric defect, and how stoichiometric defect could be utilized for multitude of applications in agriculture, organic waste disposal, and maybe even in water purification. This may come pretty handy, especially when you have to remove organic residues. But at this stage, just understand the fundamental concept of stoichiometric defect, and this stoichiometric defect could be exploited at the nano level at a much more easier or with lot of more ease. Now, the previous class I talked about the presence of iron pyrite in the hydrothermal vents. So, hydrothermal vents. So, as of now, while talking about hydrothermal vent, if we understand there are two ways life has formed. The original understanding was that life has evolved on the floor of earth, in the ocean, from the water, and the process which has governed much of it is sun. The energy, the major source of energy is sun, and living system in the form of plants have trapped solar energy in the form of photosynthesis. Photo means light, and synthesis of food using light energy, synthesis of food, or in other words synthesis of glucose molecule. Okay. So, solar energy driven synthesis of food by the plants or carbohydrates, and these carbohydrates or the plants are consumed by herbivores. Herbivores consumed by carnivores or omnivores, and then again they become part of the organic matter, and a simple cycle continues, what we call as food chain. Okay. Now, this was the only thing which is known about formation of life in terms of survival of life in terms of photosynthesis. But the storyline changed some, I would say 30-40 years back, almost 4 decades back now, during late 70s. It was observed deep inside the ocean, fairly deep in different Atlantic and Pacific rims. As these divers were going down, almost close to the floor or sea floor, they observed something which they observed a lot of smoke coming out from deep inside the ocean, and this is where there is absolutely no sunlight. And around these smokers, they name these smokers as white and black smokers. Around these smoking chimneys, in and around, it was filled with some strange life forms, and these were growing, these were bubbling all over the place. Very, very strange life forms. Now, the question which absolutely boggled the scientist is, what is the source of energy? Energy source. Because all our textbook up to that point was relying on the fact that sun is the only source of energy. Okay. But then this is a very different situation. Here there is no sun, no sun rays really can penetrate through this. Yet this place is filled with life. So who is giving energy to them? And this whole area is rich in different kind of transition metal sulfides, and the one which is in excess amount is pyrite. So, is pyrite the source of energy, or is it playing a critical role? So, stoichiometric defect, the first fact I gave you. The second fact, what I am holding out here after telling you, is the metal sulfides in hydrothermal vents. Okay, we'll pull all these threads together soon. So the third in the same line I'm going to put across is, if you look at the cell membrane, all of you have seen the cell membrane, any cell membrane, plant cell membrane, animal cell membrane, you will observe something very interesting. If these are the lipid bilayers which is forming the cell membrane, then you will see lot of proteins which are present there, like this, several proteins. So just to give you an idea, okay, so this is what you are looking at. This is a cell, and the box is the one which I am amplifying out here. And this is the bilipid membrane, and here is a magnification of bilipid membrane with polar head groups. And polar head groups are here also on both inside and outside, and with the hydrophobic tail. And here you are having these array of membrane protein which are sitting there. They could be channels, they could be pore, and series of them are there, especially in the chloroplast, mitochondria, and in their membrane, which are also lipid bilayer membrane. There are lot of iron sulfur clusters, as if, and these iron sulfur clusters are involved in electron transfer, whether it is in mitochondria, whether it is in chloroplast, or it is mitochondria. Now, if you look at it, it seems like life has evolved around iron and sulfur in the most strange places. If you look at it, hydrothermal vent, which is one of the most strange place where life has evolved, and there was a name which was given to the life form which evolved there, which is not dependent on light. So independent of light, there is no light, and these kind of life forms which evolved there is called, the process is called chemosynthesis, or the energy for life form to survive is derived from some kind of a chemical, some kind of a breaking of the chemical. Okay. So clue number 1, what I gave you is the stoichiometric defect 1. Second, the life form in the hydrothermal vents 2, and this part is filled with the FeS2. What you observe here, as I told you, is nano FeS2 or nano pyrite. Okay. And the third thing is, most of our current day cells have lot of this iron sulfur clusters which are involved in short and long range electron transport. Now, the remark we are making looks like iron sulfur complexes or iron sulfur compounds plays a critical role in our day-to-day life. Now, getting inspired by this, during 2010, 2011, we attempted a very simple experiments, and that's what we're going to share now. So, natural pyrite comes, the bulk natural pyrites comes with the contaminants of lot of different other atoms, because it has profound binding with the sulfur atoms, and you will see cadmium, arsenic and all these kind of impurities. So, what we did, we synthesize nanopyrites. So, how we synthesize? So, as I told you, there are three things which you will follow. We will talk about the synthesis, we will talk about characterization, and in that process we will figure out what is the shape and the dynamics of the molecule. So, we synthesize nanopyrite by a simple way. We made polysulfur, and I will give you the references. In one tube we made a lot of polysulfur molecules. Polysulfur means you have S2, S4, S6 likewise. Okay. It is a mixture which is called polysulfide. Okay. We made a lot of polysulfides in one vessel, and in the other vessel we have iron salt to react, and this reaction happen in an inert environment where we allow polysulfide to react with iron salt. Okay. And this is one of the process of bottom up approach of synthesizing FeS2 nanoparticles, and in an inert environment, organized environment, this reaction happen. The reason to have an organized environment is otherwise iron will get oxidized to Fe3 state. So, when we talk about FeS2 or FeS, these are the moieties where iron is in oxidation state of +2. Otherwise, if you look in any iron compounds in the floor of earth, they all are oxidized to Fe2 to Fe3 in the presence of oxygen, and what we call as, which leads to the formation of rusting. So, here we are having certain iron on the crust of earth or on the earth floor which maintain their oxidation state in 2, and same here, most of the iron sulfur cluster or all the iron sulfur clusters, iron is in an oxidation state of +2. They are in the ferrous state. Same out here, here iron is in oxidation state of +2, whether it is hydrothermal vent. Okay. Now, I will provide the detailed layout of how we made the iron sulfide. So, but this is the overall synthesis of iron sulfide by a bottom up approach, where in a small round bottom flask you created an organized environment, you created an minus oxygen environment, and at moderately low temperature of around 40 degree centigrade to 80 degree centigrade, you synthesize iron sulfide. Next, what we tried, we thought that could this agent be used as a seed stimulant, because if iron sulfur has been involved in the evolution of or the survival of life, could it do some very interesting stuff to the seeds? It is a very out of the box, and just let us give it a shot. So, what we did is, instead of, because this was clear that you cannot really apply this to the field because it may lead to acidity and all sorts of other problems, but seed treatment, if you recollect in the very beginning when we started off, yes, the seed treatment or seed pre-treatment agent. So, this is where we are starting to do a seed pre-treatment. So, what you do, you take a vessel. You have all your seeds here, and you put water in them, and on that you add a finite amount of FeS2, nano FeS2. Nano FeS2 does not dissolve in water. So what it forms is an aqueous suspension. So what you see is, if you really sonicate it a whole lot, you will see suspensions. Its sizes may even go further smaller, but there will be suspension all over the place. And to start off with, some of our initial experiments were with chickpea, spinach, rice, to see what is the effect of nano FeS2 as a seed treatment agent. So we'll continue the story from here, what we observed and what is the research which have undergone since, I would say, 2011 till 2018, when I am delivering this course, on different crops, effect of a nanomaterial as a seed priming or as a seed treatment agent. Okay, thank you.